Welcome, everyone. This is the last session of the 6th International Symposium on Intrauterine Devices for Women's Health. This is virtual event 6, and this is an exciting one, and perhaps the best one to end on are the emerging technologies that are the new methods for the future and identifying the next 10 years. So the faculty, you can read here, myself, Kate Curtis, Allison Edelman, Lisa Haddad, Sadia, Haider, Regine Sitruk-Ware is standing in for Vivian Brache graciously, and David Turok and Kim Woodrow. The agenda, I'll read these as we introduce each topic. Go forward. Learning objectives. The 6th International Symposium will provide a focused and dedicated forum for researchers, clinicians, product developers, and advocates to gain a thorough understanding of the latest science in IUD development and practice, identify gaps in research and product development, learn about innovative efforts to increase utilization of this key contraceptive technology, and foster a new generation of clinicians, researchers, and product developers for efforts. At the conclusion of this webinar, well, you can read what that one was. And if you need accreditation, this is possible through the Columbia University system. You have to complete the course evaluation at the end of the webinar in order to get credit. And this disclosures for the faculty, I'm not going to read them, but you can see what they might be. Okay, this is acknowledgements of Bayer Healthcare Pharmaceuticals, Cooper Surgical, William and Flora Hewitt Foundation, and special thanks to Mona Lisa and Sabila Pharmaceuticals. I'm going to introduce the first speaker for this morning is David Turok from the University of Utah. And the title is A Novel Platform Design for Copper and LNG IUDs, LNG being levonorgestrel. David, take it away.
Thank you very much, Diana, and thank you to the IUD Symposium. I think this is the first conference that I've ever been to that's spanned over a year, which is awesome, and I think demonstrative of the dedication that this group has to finding new and better solutions to people's contraceptiveness. And one of the beauties of Zoom meetings is they're generally super easy to get to. This is, though, I think the longest journey I've ever had to a meeting, because I started yesterday at this time rowing a boat on the Salmon River, and so a combination of rowing, walking, and driving. I got here this morning. So anyway, it's a pleasure to be here, and thank you very much for the opportunity. I'm going to talk about two products that are currently undergoing the studies for FDA approval, and I'll let you know where they're at. I will refer to them as the novel copper nitinol copper IUD and novel copper nitinol levonorgestrel IUD. They're both produced by a company called Sabella Pharmaceuticals, and their names, the copper device is otherwise known as Vericept, and the levonorgestrel device is otherwise known as Levocept. And I serve as a consultant for that same company, and our department receives research funding from a variety of other pharmaceutical companies. Next, please. So you have, if you are a clinician, you may have noticed that in the United States, we do not have a single device of copper and levonorgestrel IUD that have the same inserter yet. These things exist out in the world, right? Like the most one of the most basic IUD designs, the Nova T. That shape and that inserter style comes available in a insertion method for the two, and is available in certain countries in Europe and potentially Canada at sometimes. Anyway, what if though we had this in the United States, and what if it wasn't just some old design? And Gynafix is also a design that's approved in Europe that has both available, though not as widely used. But what if we had a new platform that actually brought some real novel attributes? What if the device came preloaded so that you didn't have to mess with getting it in the insertion tube? What if it was a very narrow insertion tube and facilitated placement, and had a soft, tapered, rounded tip, and had pre-cut strings? All of those things could make IUD placement simpler. Next, please. Particularly for the copper IUD, we are using a method, thanks to the Population Council's work in the late 70s and early 80s. But something, the current T380A, the Paragard IUD, was FDA approved in 1984. And if technology is your thing, you may recall that in 1984, pushing the leading edge of technology was the Macintosh computer. There's people who weren't probably ever on this call who weren't even born then, but it was a single box that had the hard drive. You put in that hard disk and the screen all in one. And nobody's logging into this today on a Macintosh computer. It's we've made progress in lots of other ways in society, and it seems a reasonable thing. We would make some more progress in intrauterine contraception. That design, which is, without improvements in the last 35 years, has some limitations. Among IUDs, when you fold the ends of the T for the T380A, it's got the widest diameter, really, of all the FDA-approved IUDs. It's there have been an increasing, I think, awareness of parts of that plastic frame remaining in the myometrium after long use and after IUD removal. And the copper load may be associated with increased bleeding and cramping, and there's certainly an opportunity to decrease that. Next, please. And, thinking globally, there's an increase in intrauterine contraception. There's definitely a move toward greater interest in non-hormonal methods. So what if there was a copper method that came in a frame that could be easily compacted into a narrower insertion tube, came preloaded with the pre-cut strings, and the copper quantity was less than half? Theoretically, that might make for less bleeding and cramping. And this could work regarding efficacy, because the copper would be placed at the, I'm not sure, are you able to see my cursor on the screen?
I can't see it, Dave.
Okay.
Do you want me to point something out for you?
Sure. Yes. At the base of the copper IUD, there's a copper
I can see it.
Okay. Oh. And then up by the cornu, bilaterally, there's additional copper. So it's about the placement, not about the total quantity. And then on that same frame design with the same inserter, you could also put a sleeve of levonorgestrel that had 52 milligrams, as we've got FDA-approved devices for that in the US. So what the rest of this is going to be is just an update on where we're at with these products and some of their clinical attributes. And the bottom line is the copper device has gone through, completed the phase 2 trial and has completed enrollment in about half of the follow-up or more. in the phase 3 trial. And then the leave-in adjustable device has completed the phase 2 trial and is just about to start the phase 3 trial. And then this is a picture, obviously, of both devices, as well as that insertion, the inserter tube. Okay, next, please. And here's just some other images. This is what the copper device looks like, excuse me, placed inside the tube, and the diameter is 3.7 millimeters. And then, a characteristic of nitinol is its memory. So the shape that it is created, no matter how you bend it, it will go back to that initial shape. And the flexibility in the frame allows intrauterine placement, pictured to the right, allows the top portions of the IUD to really adjust and adapt to any intrauterine dimensions, which would theoretically keep the copper concentration high at the cornu and at the cervix, where the two critical pathway points for sperm. Okay, next, please. All right, so the phase 2 data, as I mentioned, this has already been completed. It had 286 participants who were followed for 36 months, and this is published the outcomes are published in the Green Journal. And the primary endpoint was 12-month efficacy by Pearl Index, and there were secondary outcomes that you'll hear again in this presentation, but study drug placement, safety, tolerability. Next, please. And then this is some of the demographics of the participants. And over 90% of the participants in all of these studies, these 3 studies I'm going to talk about, were evaluable for pregnancy. So essentially efficacy data, and then a smaller portion were older people who were 35 and up, who were included for safety data. And anyway, this is just some baseline characteristics, and age of 27, racial and ethnic mix, not too far off the national representation. The majority of people were nulligravid and even greater nulliparous, and then some other attributes. And then next, please. And just looking at the placement data, essentially 99% of people had successful placement. Some of those took a second attempt. Mean pain score in placement for a 1 to 5 Likert scale was 2.4. Perforations, there were none. And the cumulative expulsion rates have been quite low. And even, almost basically 1% at 1 year. And the Pearl Index at 1 year here was 0.5. And that image is an ultrasound image where you can see the copper pledgets on the frame in the uterus. Next, please. And then this is just a slide life table and a KM curve for discontinuation over 36 months. And that dotted line at the top is for subjects who had device expulsion, so very small proportion of discontinuers. At 12 months, we had almost 80% of people continuing, and we offered, the initial study was to be 12 months, and then people were offered continuation. So there's a bit longer horizontal section here at 12 months where the larger group discontinued than at other times. Next. And those data supported initiation of the phase 3 trial, which, as I'd mentioned, enrollment has already been complete. There's 1,620 people at 42 sites. Primary endpoint is Pearl Index at 1 through 3 years. We'll achieve the 3-year mark in next August, and same secondary outcomes. Next, please. And the data at the 1 year mark were recently presented by Mitch Cranon and company at, and some other providers at different sites at the ACOG, the recent ACOG meeting. And the first column demonstrates the demographic characteristics similar to that preceding slide. And then this is the efficacy for pregnancy population less than or equal to 35 years. And this is the older group that are the safety population. And again, mixes are similar. There's Black or African American people represent 15% of the population, and 19% are Hispanic or Latinx. The nulliparous group is obviously we would expect to be larger in the efficacy in the younger group, as we see. And then the BMI was less than 30 for the majority, but a significant portion of people were greater than that. Next, please. So the outcome data on IUD placement with first placement was basically 96%. It went up to almost to 99% after a second try. There were 2 perforations, 0.1%, similar to other IUD studies, and cumulative expulsions here have been a little bit higher, 2.3%. And there has been, we haven't completely analyzed this, but as there's been more people using recyclable menstrual products, cups, and it does seem like they're, and you know, if you think about people who don't want hormones might also be more interested in using non-disposable menstrual products, and there's an intersection of those two things, and it does seem that it's possible to remove the device and the cup simultaneously. Next, please. And then this is just, these data are showing the Pearl Index. So there's 9 pregnancies in the first year, Pearl Index of 0.9, and the mean number of bleeding and spotting days per cycle decreases abruptly, most in the first cycle, and then cycles 3 through the rest of that first year return to essentially baseline. Next, please. And then, so those are the copper IUD studies, phase 2 and phase 3. The levonorgestrel 52 milligram nitinol IUD has completed the phase 2 study, and there will be a publication forthcoming on this. And some of these data have been presented at national meetings. Year 3 was completed in June 2020. And again, this is what the device looks like. It's got that nitinol frame with a levonorgestrel sleeve on it. Next, please. And we had the ability to compare the phase 2 data between the copper and the levonorgestrel product. Similar sample sizes, similar success with device deployment on first and second attempt, and the ease of device placement. It's also similar, and then the cumulative expulsions, 1% in the copper group at 2.5 in the levo group. That's next, please. And in summary, there are favorable attributes of these products and favorable data to support their continued progress through the FDA approval process. For the copper nitinol device, less copper, narrow insertion tube, preloaded device, pre-cut strings, favorable data, less insertion pain from a prior IUD study, fewer expulsions when we compare the data that we have from these products, the nitinol copper IUD to the FDA approval data from Paragard, and it appears lower discontinuation rates. And there's a favorable path to approval with phase 2 completed and the first 2 years of phase 3 completed now. For the levonorgestrel 52 milligram product, we're using an approach that's time tested as far as the quantity and the delivery system for the levonorgestrel, preloaded, pre-cut strings, no pregnancies in phase 2, and the same drug and dose is approved by those 2 other US products. And again, it appears to be on a favorable path to approval with completed phase 2 data and soon to be initiating phase 3. So I will say thank you very much for your interest in this, and look forward to hearing the other speakers. There's lots more interesting information to come. Thank you very much for the opportunity.
If anyone wants to ask questions, they can enter it in the chat. I will ask a question. Did you warn women or advise women against using menstrual cups throughout the study or at all?
Eventually, we did, and there was, we had initially we weren't aware of this, and it became, as we became aware of it, we added information about that to all of the interactions. And I think, my guess is the majority of people who use the product, a menstrual cup or catchment, or if it's placed distally in the vagina, close to the introitus, so not up against the cervix, and when it's removed, it's pinched initially to release pressure and not create suction. I think the majority of the people who are using them use them, and it continues to work and stay in place just fine. But I think if it's removed quickly and it's placed high, my sense is that's where it's happening.
Are you tracking menstrual cup use in participants now, or whatever hygiene products they may be using?
We have some data on that.
Okay. There are questions. So Mitch Crennan asks, Dave, expulsion is hard to compare with old data because expulsion is parity dependent.
Yeah. So this is, Mitch, excellent point. But Matt Reeves led a team that did an RCT in the Dominican Republic prior to these studies. That was an RCT of the Varicept versus the copper T380S. And I'm sorry, I did not present those data. They were published in Contraception. And basically what that showed was lower expulsion rates when they were compared head to head. So we do have some head-to-head data.
The next question is, to what length are the strings pre-cut, and what are the chances strings are too short in the deep uterine cavities?
That's a great question. So it's a little over 10 centimeters total. And I have not seen cases where the strings are not present at all. And I think there was an earlier question too about about the option for self-removal with pre-cut strings, and is that going to change things? Yeah, I mean, I don't know what the number is in the bell-shaped curve, what the top end is for uterine cavity depth, but I suspect that it's quite rare that the string be adequate or will be an issue. It gives you, certainly you have the option to make it shorter, but you can't make it longer. So that could be a future issue for people.
Next question is, did you expect less bleeding with this copper IUD because the number of bleeding days seems exactly the same as the copper T380?
Right. So we do expect fewer days, with under the hypothesis that less copper will be less irritating and cause less bleeding. And we did find that when we compared directly. And then to Mitch's follow-up point there about the expulsion rates, they had a different expulsion definition in the RCT, whereas any portion of the device that entered the cervix was considered as an expulsion. So the expulsion rates there were higher overall, but higher for Paragard than they were for or the T380 versus the copper nitinol device.
Diana, just to allow more time for the speakers, why don't we switch to Dr. Woodrow? And I encourage everybody to continue putting their questions in the chat. We hope to have time to address those at the end of this presentation and then also at the end of the day. And as well, Dr. Turok can also answer some of the questions in the chat if he feels like he can represent those in writing. So sorry to interrupt.
Yep, no problem. Great, thank you. Okay, so the next talk is Kim Woodrow, and the title is Concepts for IUD Functional Integrations to Expand Applications in Women's Health. Kim?
Thank you, Diana, and thank you to the organizers and the audience this morning. I welcome the opportunity to present to a new audience. So as Diana mentioned, I am a faculty in bioengineering here at the University of Washington in Seattle. So I'm going to be sharing some of the work that we've been doing for the last several years that involve the copper IUD that David just described in great detail. So I'm really fortunate to go after him and take advantage of the additional time I get from not needing to go through a lot of the details of the value of the copper IUD. Next slide, please. So my talk today is really focused on extending the applications of potentially extending the applications of the IUD to target unmet needs in women's health, where it can have potentially unique applications. And so I'm going to share some of the work that we've been doing specifically in non-surgical permanent contraception with Dr. Jeff Jensen at OHSU as part of his permanent contraception program there. And I'm also going to share some of the work that we've been doing related to designing the IUD to incorporate antiretroviral drugs for the application of multi-purpose prevention of unintended pregnancies, as well as providing HIV prevention. I want to end with some summaries of our findings and some of the challenges that we face, but also future directions related to key scientific and engineering questions that might shape a better understanding of potential applications of the IUD in women's health. Next slide, please. So what is the need? So I'll talk about the need in regards to the specific examples that I'll share with you today. But I think, if we look at sort of the, expanded need of gynecological and reproductive health issues that face women across their lifespan, we're thinking about opportunities in which the intrauterine device, because of its design and its placement and other strengths of its use, might be able to address some of the other clinical unmet needs in women's health, such as endometriosis, uterine fibroids, gynecological cancer, and other clinical issues listed here. Next slide, please. So why an intrauterine device? So I think that the IUD, and I probably don't have to go through a lot of this for this audience, has a lot of strengths that can be leveraged for these other applications. It has extensive history of development, as David mentioned, a wealth of clinical data on safety and contraceptive efficacy. There's been a lot of studies, evidence-based models that show if you can increase access and eliminate initiation barriers and address some of the safety concerns, IUDs are actually preferred by end users over other contraceptive methods. So pills, rings, patches for contraceptive use. And we know that the worldwide use of IUDs has steadily increased. It's the most widely used method for reversible contraception amongst reproductive age women globally. Next slide, please. So David addressed a lot of the form factors related to the Varicept IUD, and this is the IUD that my group has been working with over the last few years, starting with a collaboration with Jeff Jensen. And so that's the initial work that I'll talk about. So David talked a lot about the dimensions and some of the attributes in regards to the clinical outcomes that were being addressed in those phase 2-3 studies, some of the discussion points about insertion, expulsion, bleeding, and other things. So the specific attributes that I want to reemphasize in regards to the work that I'll be sharing today is this component of the flexible shape memory frame. So this is the Nitinol frame that David mentioned that has this shape memory function in which it's lowest energy state really sort of motivates its positioning in the cornu, and that'll be important for some of the specific application I'll share in permanent contraception. This is a smaller profile frame relative to say the T380, and another component is the lower copper dose that's associated with this frame, which has implications for bleeding and such. But for the perspective of designing and extending the functionality of the IUD for other applications, from an engineering perspective, this is a strength for us because we see this as opportunities to be able to integrate other functionalities into this frame. And so from conversations with Jeff and others during our time working with the Varicept IUD, we know that potentially this low profile frame could increase in width by 50% and still be consistent with the currently available copper IUD, meaning that we can integrate and continue to design with this lower profile frame to accommodate some of our drug integrations. Next slide, please. So the first work that I'm going to talk about is this collaboration with Jeff Jensen at OHSU for applications in non-surgical permanent contraception. And so the really the goal of this project was to utilize this fundus-seeking property of the IUD, specifically the placement of the Nitinol IUD arms in the cornu as a mechanism for local delivery of sclerosing agents to that specific site to cause tubal occlusion. And so the motivation for this work obviously is that you know, approximately a quarter of reproductive age women between 15 and 49 years worldwide select permanent contraception as a form of birth control. But currently, the high demand for permanent birth control is met entirely by surgical intervention, which is a barrier in regards to access to healthcare in low middle income countries. So approximately 5 billion people, for example, worldwide lack access to safe and affordable surgical care. And so Jeff has, as part of his permanent contraceptive program, been looking at strategies to block the fallopian tube non-surgically to increase access to permanent contraception and address current unmet needs in family planning. Next slide, please. So the first question in regards to thinking about the premise and feasibility of redesigning an intrauterine device for non-surgical permanent contraception is really about, you know, the context of drug delivery and tissue exposure, so PKPD of these sclerosing agents that we intended to deliver. And so the premise for this work is really motivated through what we know in regards to natural tubal occlusion that occurs from sexually transmitted pathogens such as gonorrhea and Trichomonas. And they really illustrate the principle that, you know, can be potentially recapitulated in this particular design. So in regards to, this infection that leads to tubal occlusion, we know that for gonorrhea, it's capable of causing tubal occlusion after even just a single infection due to the potency of the oligosaccharides toxins that are released. So in contrast with Trichomonas, we know that the induced tissue damage that's caused by infection there, total epithelial delamination doesn't occur right away after the first infection, and that multiple infections are required for occlusion and scarring to occur. So related to that pathology, what this suggests to us is that, you know, the potency of an antagonistic agent can compensate for the duration of exposure, right? So gonorrhea versus Trichomonas in regards to the number of infections that lead to tubal occlusion. Finally, you know, other sort of methods for sterilization, so such as cautery or cryosurgery, chemical sterilization, and eSure have provided us really good information in regards to the correlates of exposure time that lead to the events required to occlude the fallopian tube. And then finally, work in the field related to the installation of liquid sclerosants really defined for us that, this targeted placement of these sclerosing agents within the cornu region and the enhanced residence time is really needed for efficacy of such a strategy. And so we know with liquid installation of sclerosants that less than 5% of the dose remains at the target site over time. And so from a collection of all of this data, we've been focusing on tubal occlusion strategies that lead to an initial extensive epithelial damage, so 0.4, followed by chronic inflammation that resolves in pathological scarring in lieu of reepithelialization. And so the idea is to use this Varicept IUD where each arm is integrated with a solid dosage form for local delivery of sclerosing agents that can elicit an initial acute trauma as well as sustained inflammation to provide permanent occlusion of the fallopian tubes. Next slide, please. So one of the concepts that we pursued with Jeff and has recently been published was to integrate a drug delivery system onto the arms of the Nitinol IUD. In this case, you could see this in the bottom figure of panel C. And the drug delivery system there are, it looks essentially like a Teflon tape wrapped on the arms. Thanks, Allison, for circling that. And so this is the design, this is the concept prototype that we developed with Jeff to test feasibility of this idea. We looked at several sclerosing agents that have been evaluated in the literature, so doxycycline, silver nitrate, polydocanol, and the process for the integration was to use a platform developed in my lab that formulates these sclerosants into polymeric fibers that can be easily wrapped and integrated into those arms, so they easily conform to that shape. Next slide, please. So I think one of the challenges in the field for permanent contraception and identifying sort of iterating through the pipeline of agents that are amenable to this application are really the models to do that in. And so as part of this research, we used actually a mouse model, a subcutaneous mouse model, to iterate through properties of dose, like I had mentioned in sort of the premise and feasibility slide. So thinking about dose and timing and sclerosing agent, right? And so you think about that design space and trying to identify a lead compound that we could then really validate in the baboon models, which are the standard for the field. And so this data is really showing our attempts to do that in a subcutaneous space, which is has obviously really clear limitations in regards to predicting what happens in the uterine space. But that's what we did here. We looked at inflammatory markers shown in the bar graphs on the left. We also looked at in the H&E images in the center specific. Obviously, the outcome related to fibrous capsule formation around these delivery systems that were these fibers delivering these sclerosing agents, and we quantified this as much as we can in regards to inflammatory infiltrate. And so this is the last column, and collagen deposition. And ultimately, we chose the agent in the bottom left, the silver nitrate. I'm not sure of the H&E, so it's the SN, it's letter M here, because of these primary outcomes that we measured. So next slide, please. So using this lead that came out of our subcutaneous and some of our in vitro screening, we integrated this silver nitrate containing fibrous material onto the nitinol frame and placed these into non-human primate models, so baboons by transcervical placement. So you could see a diagram of our intended function, and then the images in B and C show fluoroscopic images of fiber-loaded IUDs placed in the baboons, where we have active fibers in B compared to just the nitinol frame without the fibers. So ultimately, these images indicated to us that, the presence of these fibers on the IUD didn't preclude their placement in and it appeared well tolerated in the animals. So the H&E images to the right were really showing our primary outcomes related to occlusion of the fallopian tubes, or where we were really looking for fibrous capsule formation. And overall, we didn't see that in any of the animals that we looked at. And so, in general, there's this tissue is highly dominated by collagen, but specifically, we weren't able to observe any tubal occlusion. A good outcome of this is that they were appeared to be safe and well tolerated, but in the context of permanent contraception, that wasn't something that we observed. So next slide, please. I'm getting a little close to my time, so I'll quickly go over some of this last part on the HIV pretty fast. But one of the things that I wanted to point out were, just some of the limitations to the screening that I mentioned that we did in the subcutaneous model, and then other sort of areas where we've been looking to identify more potent sclerosant agents and really achieving this sort of acute response that I think is really necessary to cause that provocation needed for a tubal occlusion. So next slide, please. So really quickly, my lab, just to switch to our final application of the IUD, has been looking at developing the nitinol frame for multi-purpose prevention, HIV prevention, as well as long-acting contraception. So I won't go over this. Next slide, please. We think that there is a lot of premise and feasibility for achieving long-acting HIV prevention integration into this intrauterine device. There's a lot of data from the HIV prevention field, specifically regards to the recent clinical study of the dapivirine ring that really allows us to think about formulations that really have to deliver a potent enough compound to get enough sort of drug delivery in a single sort of device for at least a year, and hopefully out to 3 years for HIV prevention. We think what that is less than 50 mg total drug releasing at a daily rate of 10 micrograms. And so that's what we've been looking for. Next slide, please. So we have a recently funded NIH grant. A lot of this original work on MPTs came from programs initiated by Diana and others at NIH. We're funded for device that is shown here, mirroring sort of the copper IUD Varicept design with the Levacept design, and we're actually looking at drug integrations for HIV, looking at the historical reservoir drug delivery system. So this is what's delivering those hormones in the LNG based designs, and then matrix drug delivery systems, just like I had shown with what we've been doing for the permanent contraception. Next slide, please. So we have data from my lab. I'm not showing it here. It's not published yet, showing that these drug delivery systems that are delivering these antiretrovirals are safe and well tolerated vaginally. They show rapid onset and sustained concentrations in cervical vaginal secretions and tissues when administered as a single dose in non-human primates topically to the vaginal vault, and that we're able to detect drug depots in both the tissue and the drug delivery system after a period of time. And so we think that this strongly promises some of the work that we're proposing for IUD integration. Next slide, please. So some of the properties that we think are particularly interesting for these matrix drug delivery systems is this ability to provide kind of a tissue and cellular depot that's based on some of the muco-adhesive properties of the fibers, and the ability of some of these formulations to lend themselves to amorphous solid drug dispersion. So specific properties that can really formulate these highly potent compounds used for being used for antiretroviral efficacy. I think some of the drug dispersion properties that we're looking at to extend the long-acting potential are related to the nanocrystals themselves, and so that's a big focus of my lab. And the challenge really is to kind of combine these attributes into a formulation that meets a minimum standard of HIV prevention for at least 12 months. Obviously, the contraceptive piece of it is already, well established in regards to the Varicept. So we aren't changing anything about that, and just trying to integrate that HIV prevention component. Next slide, please.
You need to wrap up, Kim. It's a half hour.
Okay, so in summary, I won't go over these specific points, but I think the remaining questions for us in regards to these other applications that we're interested in is really thinking about local and systemic PK/PD of these drugs, and so that's a major factor for these ARV drugs integrating into the IUD. So what is the potential safety, toxicity, and issues related to these antiretrovirals delivered locally, identifying APIs with a proper therapeutic index in regards to, for example, the sclerostin agents for permanent contraception, and potentially delivery of combination agents for combination applications for multi-purpose prevention. So with that, I will end with my last slide, and thank you all for being here and this opportunity to present. Thanks.
Thank you very much. In the interest of time, we'll go to the next talk, and if there are questions, please put them in the chat, and Kim will get to them hopefully later. So the next talk is proof of concept study evaluating a copper intrauterine contraceptive system, releasing 3 different doses of ulipristal acetate, and Vivian Bracci is the senior author, but Vivian is ill, and Regine has offered to step in and give the presentation. Regine.
Hey, Diana. I'm working at the Population Council, and that's my only conflict of interest as we are developing this new concept, and I'm co-author on a patent for this system. I'm really sorry that Vivian was ill, and I do hope she will recover quickly. And the four names on this slide are the people who had prepared the presentation, but there is a big team who have worked on this proof of concept, and we are very proud that the publication has been accepted very recently and is currently in press in the journal Contraception. The next slide will speak about the concept. Excuse me. So as you have heard from David, there is a rationale, of course, for improving the copper IUDs, which are very highly effective, safe, and however, it increased the amount and number of bleeding days. And the objective we had was to reduce the unfavorable bleeding and enhance its acceptability. Now, of course, the Mirena that was also developed by the Pope Council long ago and has been the model for many other levonorgestrel IUS, has answered some of the decrease in bleeding, but not the irregular spotting, which follows the insertion within the first few months. So our objective was really to try to obtain a regular bleeding. And this was based on some preliminary data we had in a study with rhesus macaque in the Oregon Primate Center, where inserting a kind of implant like an IUS, delivering UPA in monkeys who had also estradiol and progesterone implant to reproduce a cycle, there was an endometrial atrophy and a complete reduction in bleeding and amenorrhea in most of the macaques. So based on this result, we developed the concept that if we developed an IUS that would deliver the copper for ensuring the well-established contraceptive efficacy and add on a very small dose of UD acetate that we know may decrease the amount of bleeding and acts on the endometrium, and at a low dose that would not interfere with ovulation, we may have, in fact, less risks. And in the next slide, which continues on the concept, we had on previous study with another delivery system with a vaginal ring delivering ulipristal acetate, we noted that the changes in the endometrium that were named progesterone receptor modulator associated endometrial changes, PAECs, as well as heavy bleeding, were dramatically reduced when ovulation was maintained. Possibly the progesterone secretion during this phase of the cycle that results in menstrual shedding may prevent or reduce the frequency of this event, or the PAEC, which is the main difficulty we face when using ulipristal acetate. So that was the hypothesis. Will small dose of UPA administered directly in an IUS result in reduced bleeding without ovulation inhibition and without endometrial modification? So we designed this proof of concept, which was the first proof of concept with this system, which is a prototype. Next slide, please. And the primary objective of this study was to assess the pharmacodynamic outcomes, of course, having in this IUS 200 square millimeter of copper, which is rather low compared to the 380A, slightly higher than the Veracept you have heard about, but it's well known that it's very effective, and these were the very old studies of WHO demonstrated its efficacy. And we wanted to add different doses 5, 20, or 40 microgram per day of delivering the UPA over 12 weeks treatment. So the system, the prototype in the reservoir, and I will show you the design later, was in fact with the goal of at least a 3 years delivery system and maybe a 5 years delivery system. But for the purpose of the study, we had the prototype for a short 12-week study just to demonstrate that our hypothesis was realistic. So we would measure the bleeding pattern, essentially, the follicle development and ovulation, and of course, overall safety endpoints, including the endometrium. And Alistair Williams in Edinburgh reviewed all the biopsies for PAC changes. Ultrasound measured endometrial thickness, and then, of course, record of adverse event, liver tests, and pharmacokinetic profile of UPA plus tissue concentration of UPA in the biopsies. So we had a sample size calculated on the basis of a relative difference of 40% in the number of bleeding and spotting days between baseline and end of 3 cycles of treatment, based on the fact that we know the copper IUD would have increased the number of bleeding and spotting days by 41.7% at 3 months, and this has been published, it is well known. So we had to get at least, well, 8 participants per group to complete the study. And we had, in fact, enrolled 29, which is 10 per group, less 1. Next slide. You see here the picture of this prototype. So it's really based on the copper T standard, and it was produced in a factory in India that produced both the levonorgestrel IUD and the copper IUD, sold via UNFPA, and you know they had really a very strong platform and good manufacturing practice. And the concept we had was to add the copper around the horizontal arms and the first upper part of the vertical arm, and then the end of the vertical arm was the reservoir that would have a total load up to 36 milligram of UPA, but to deliver micrograms per day, 5, 20, or 40 microgram per day. So 30 women were randomized at the Pro Familia clinic of Vivian Brash, and the recruitment started in May 2017, and then the study was completed with 29 women enrolled. Next slide showed you the study design, where we had, of course, a lead-in cycle, control cycle, to demonstrate that these women ovulate. And then they had the insertion of the IUS on day 5 to 8 of the first cycle. And it was kept for 12 weeks, then removed. And then there was 2 bleeding episodes as a recovery cycle. And the thick arrows that you see are the insertion of the, the time of the biopsies. And the endometrial biopsies were done at initiation in the follicular phase of the baseline cycle, then at the end of the 12 weeks, and then at recovery. The next slide will show the population, where in the 3 groups we had similar age, weight, and BMI. And we had confirmation that all the 29 participants had ovulation in the baseline cycle. The next slide shows you the results in number of days of bleeding. And you have the 3 groups, 5 microgram, 20 microgram, and 40 microgram. The orange bar is the pre the baseline information. The light green is the treatment cycle 1. Then in light blue, treatment cycle 2, and dark blue is treatment cycle 3. So you could see in the low dose, the 5 microgram, that there was a slight increase in the number of bleeding on the first cycle, and then it decreased. No, sorry, could you stay on that slide before the previous? Yes. And on the 20 microgram, you see a decrease on cycle 2, cycle 3, and it is significant statistically only with the 40 microgram per day, where we had really suppression of the decrease of the number of days of bleeding with a p-value below 0.05. But there was a trend also in the 20 microgram group that didn't reach significance. And we could see why in the next slide. And the next slide is a waterfall representation. And the waterfall plot was one of the statistical tests that was made by our TITS fellow who worked on this analysis together with Carol Sales, Maud Lancio. And here you can see each subject, and the bar represents the percentage of change in the number of bleeding and spotting day from baseline to treatment cycle 3 for each of them, according to the dose. You have in red the 5, in green the 20, and in blue the 40 microgram per day. You could see that one patient increased considerably in the 5 microgram per day, which might explain why there was, as a median, no change. The 20 microgram, you can see that many women had a decrease. Well, many, 7, and 3 of them had a slight increase. While on the 40 microgram per day, only 1 had a slight increase, and all of them decreased the number of days. Then on the next slide, you can see the summary about the ovulation and the ovarian function. And the definitions are in on the left lower panel, where you see that ovulation is the follicle ruptured, followed by an elevation of progesterone above 3 nanogram per ml. We had definitions of LUF, luteinized and ruptured follicle, where there was no rupture, but an increase in progesterone above 2 nanogram per ml. And a category named follicular development without resolution, which means that during each 4 weeks considered as a cycle, we didn't have a rupture, and therefore the events occurred in the next cycle. So we have to calculate over the 4-week cycles what were the events in the follicle development. So in the 5 microgram dose, 96% of women ovulated. There was no LUF, and only 1 cycle had a follicular development without resolution. In the 20 microgram dose, you can see 87% of ovulation. And in fact, the 10% is due to the follicular development without resolution, 3 women and only 1 LUF. And with the 40 microgram dose, 81% ovulated, but, well, excuse me, yes, 81% of cycles had an ovulation, 19% of cycle had LUF, and there was none in the category without resolution. And the same results are represented in bar chart below on the lower panel, where you see the ovulation was slightly prevented with a higher dose, which is still a very low dose, but it started to be already interacting with ovulation by inducing a luteinized and ruptured follicle. And Next slide shows you the effects on the endometrium. No, excuse me, the progesterone levels, which have been recorded with measures that we had twice a week. And you can see in different colors the three groups. And grossly on the curves, you can see that they were ovulation in all the cycles and recovery cycles, slightly higher levels of progesterone. The next slide would show you the endometrial histology, and that what was of concern, finding a dose where there was absolutely no changes of this PRM-associated endometrial changes, which are non-physiological as defined by Alistair Williams. And I highlighted in yellow here, you could see that in a 5 microgram, there was only one PAC, the same in the 20 microgram, one in 10 participants. And in the 40 microgram, there was 4. So here again, a sign that the dose of 40 microgram was slightly too high, although it was the only one that really reduced the number of bleedings significantly from a statistical point of view. The next slide would give you more information about the safety as far as liver is concerned. And you know that the PRM and essentially SMIA, which is uliprysyl acetate tablets of 5 milligram, approved in Europe for the treatment of fibroma, had a review of its safety due to liver, acute liver failure. And therefore, there was the necessity of checking the liver enzymes. When we started the trial, there, in Vivian's clinic, there was no such discussion yet. So we had then measured the AST and ALT for the 3 groups. It's a short study, it's only 12 weeks, but still we could see that there was no changes. And the only statistical point that are significant, if you see on the last line at 0.001, is in fact a decrease in ALT in the high dose group. Only one participant still had an increase, a minor transient increase, but that didn't reach the level of anomaly. It was below 3 times the upper limit of normal. Next slide. So in terms of adverse events, the lower abdominal pain was the most frequent, reported by the majority of women. But these complaints were reported shortly after insertion or in the first month, and they were mild or moderate, and there was no severe cases. There was a headache, bacterial vaginosis, and minor unrelated complaint, as you see, but no serious adverse event occurred during the study. Next slide shows you the pharmacokinetic profile. And if you look at the y-axis, you can see that it's a very low level. It's the levels, the median uliprysyl acetate concentration expressed in nanogram per ml. And we had a maximum of 0.2 nanogram per ml with a higher dose of 40 microgram per day. And you see in the small table, which is at the top of the graph in blue, that with the oral of 5 milligram day tablets, the level, the Cmax is 23.5 nanogram per ml, where here our maximum Cmax was 0.19. So there is a big difference, and the fact that there is a local concentration but no circulating levels could be an advantage even for the treatment of fibroma, because there would be much less in the first pass effect on the liver. The next slide will come to the conclusions, where most women had decreased bleeding with all dose, but most statistically significant only in the high dose group. All women were very satisfied or very or extremely satisfied of the pattern they had because they saw this decrease and it was regular, predictable. The ovulation occurred in most cycle, although we had LUF with the 40 microgram dose, and we had this withdrawal bleeding after progesterone decrease. We saw a bleeding in all cycles. And I think at the end, we had another conclusion where we didn't see an increase in endometrial thickness. The PAEC that we observed were more frequent in the higher dose, as I explained, and this despite the shedding, despite the progesterone bleeding withdrawal. We didn't have a safety concern regarding liver enzyme or any other adverse event, no SAE. And the 20 microgram dose also had not reached the significance, is the most promising dose because ovulation was not suppressed. We had only one LUF. The amount of bleeding decreased in general. Only 3 women had some increase. And only 1 of 10 biopsies had PAEC. So possibly the 10 microgram dose could have even better results, but we had not tested it, and it may be warranted. And with this, I think the high continuous dose of UPA that had led to safety concern had really been very damaging to the field of the PRM and the ulipristal acetate. Unfortunately, even though some cases could not be definitely related, the causal relationship was not clearly established. There was a decrease and a strict restriction of the prescription of SMIA in Europe. And maybe the intrauterine administration of this very low dose of UPA that minimize the first pass hepatic effect could be feasible, both for contraception and other indication. But because of the long-term purpose of contraception, we need long-term efficacy and safety study to establish the potential risk-benefit of this novel IUS. And to end and thank you, I wanted to show the list of and acknowledge a funding source. The study was conducted with support from a grant from NICHD and the Population Council, and also contribution of HRA Pharma France, who also contributed to the to overseeing the IUS manufacturing in India, and the clinical team at ProFamilia with Vivian Brash as the first author, and Population Council team, and Ruth, Marlena, Narendra Kumar, Mohsin Alami, and Dan Lovell. And the main collaboration we had with Carol Sales Vieira in Campinas, as she was a TITS fellow at the time. We were having the study organized, and she has supervised the study together with Ruth Merkatz. And we worked, of course, with Jeff Jensen, Alistair William, Indrani Bakshi, who did some other tests in the endometrium, and SRI in California. Thank you.
Thank you very much, Regine. And yes, put your questions into the chat. We will need to go on to the next session. Thanks very much, everyone.
Thank you, everybody. I am going to switch to our next last panel, and our panel has the great pleasure of closing out the 6th International Symposium on IUDs. I just want to give a huge thank you and round of virtual applause to the coordinating committee and Miss Rachel Ferret, who's our administrator for the meetings. They really persevered through all the challenges that COVID brought this year to scientific meetings and organized an amazing series of webinars over the last year. I'll be moderating this last session. I've had the unique position of moderating the whole thing today because of some internet difficulties, but my name is Allison Edelman. I'm a professor in OBGYN at Oregon Health and Science University in Portland, Oregon. I'm joined today by several amazing colleagues and scientists. Dr. Sadia Haider is Vice Chair of Research and Faculty Affairs at Rush University in Chicago. Dr. Kate Curtis is a health scientist with the Division of Reproductive Health at the U.S. Centers for Disease Control. And Dr. Lisa Haddad is the medical director of the Center for Biomedical Research at the Population Council. our session is focused on really the gaps and futures of and goals of IUD research. And our speakers were tasked today with reviewing the work presented throughout the year and really reflecting on how the work is still yet to be done, and including how we can improve our research methodology to integrate, respect, and elevate diverse perspectives and populations. How does this new research impact clinical guidance, and how are patient perspectives and diversity reflected in that guidance? And finally, what are the IUD gaps in research, and what might the future of research look like? So with that, I'm going to let Dr. Sadia Haider take over.
Great. Thank you so much, Allison. Thank you to the IUD Symposium and organizers, and thank you for the opportunity to speak today. So I'm going to completely shift gears from our previous speakers, and I was tasked to talk about reproductive justice, specifically as we think about informing our contraceptive research. So I'm going to take us back to the September symposium. Dr. Westhoff stated in her opening remarks for the symposium in September that this is the first time we are discussing centering the patient voice in the context of IUD research and development at this meeting over the years. And I want to bring us back to that as we conclude this meeting today, given what the patient desires is an important aspect of our research going forward. I have the opportunity to reflect, as Allison said, we are reflecting back on the sessions that have happened over the year. And I'm reflecting on the session in September that was the opening session titled Putting Patients First, the Importance of Centering Patients' Voices in Clinical Care and Research. This was presented by Dr. Jamila Perret, Dr. Liza Fuentes, and Dr. Christine Dellendorf. If you have not had a chance to view this session, I highly recommend you go back and view this recorded session as a way of framing our perspectives as we move forward in research. As we think about the future of family planning research, we need to take into account the history of reproductive health care, where we have come from and where we hope to be in terms of balancing equity with access in developing the science and technology of IUDs. The history of family planning globally is rooted in a history of eugenics and population control. I know we are all familiar with this. We know that there are countless examples of this throughout our history, in which highly educated persons of good social class were considered eugenically superior. We also know about the population control movement, which aimed to solve social and economic problems through fertility control, focused on coercive population campaigns that targeted the most powerless of people. The birth control movement followed suit and was also problematic with its focus on poverty control and its use of experimental family planning centers. And this was followed by the advent of new technologies. And globally, there were many birth control experiments and test trials that occurred in many countries without ethical processes in place. Again, our history has really stratified reproduction throughout our work, in which power inequalities between groups has really focused on reproduction and encouraged it in a differential fashion. Why is this important to take into consideration? It's really resulted in a biased and discriminatory healthcare system in which many individuals are less likely to access quality and culturally competent care and less likely to trust the healthcare system. Next slide, please. So where are we now? So there's a recognition of a need to move past this history and frame our work in reproductive justice. I know many of you are familiar with this, but I'll just review it here briefly. So reproductive justice is the intersection between social justice and reproductive rights. It was started in the 60s and 70s, primarily by women of color who were sort of left out. Their voices and perspectives were left out in the reproductive rights movement. It uses a human rights framework in which all people have the human right to be fertile, to engage in sexual relations, to reproduce or not, and to care for our children with dignity and safety. Next slide, please. So what's the difference between reproductive justice and reproductive rights? Reproductive rights is really focused on the legal rights. Much of our work has been focused on abortion rights, specifically when it comes to reproductive rights. Reproductive justice goes beyond this, and it focuses on populations who are most vulnerable to restrictions on their reproductive rights. So reproductive justice is human rights seen through the lens of the nuanced ways that oppression impacts self-determined family creation. Next slide, please. So though we have moved past our history of sort of this overt eugenics and population control, we are still on this journey towards incorporating reproductive justice principles into our practice, policies, and research. In the recent past, we've focused our efforts on increases in access to contraception through promotion of IUDs, with a real emphasis on efficacy, and really use this sort of tiered based counseling approach, highlighting what we as scientists and clinicians think is the most important aspect of the method, its efficacy. As we move forward, we need to take into account the patient perspective and whether that's what is really valued by the patient and what their experiences have been. A number of studies by our colleagues in the field highlight these perspectives and the need to continue to center patients in the work we do. So what are the patients telling us? What are their voices saying? When it comes to IUD care specifically, patients express that many factors beyond effectiveness influence method selection and continuation, and that the focus on effectiveness points to the tension between the public health goal of reducing unintended pregnancies and the individual patient level perspective and desires. Patients have felt a resistance to early IUD removal despite their own symptoms and concerns, and a refusal by providers to remove IUDs. This was discussed in detail by Dr. Liza Fuentes during the webinar in September. They've felt pressured and coerced to get a LARC method. Evidence has shown racially based discrimination counseling. Dr. Dellendorf's work has highlighted this and very clearly, and it's shown the impact where it results in greater recommendation for IUDs to socially marginalized women. Overall, this is concerning that practices that implicitly pressure women in contraceptive counseling can infringe on reproductive autonomy and reinforce health inequities when they engender mistrust of the healthcare system and contraception. Next slide. So this leads us to think about contraceptive provision and research not from an efficacy or tiered-based approach, but rather from a patient-first approach, trying to incorporate the elements that reproductive justice calls for. I again here will refer to much of the literature from our own community of family planning researchers, whose work has given us strategies for moving forward. Based on this research, there's a call to individualized counseling and really taking the patient values into consideration, conducting shared decision-making when doing contraception counseling, acknowledging our implicit bias as providers and how implicit bias serves as a barrier to reproductive autonomy and justice, recognition of biases in our use of terminology that target specific groups of people. And this is often how we've done our research. We've used terms like Medicaid, at-risk, uninsured, underserved in our work. And there's a bias that underlies these terms. Acknowledging that there's statistical discrimination. So what does this mean? It means using epidemiological data or previous clinical experience to estimate a particular woman's risk without consideration of the patient's unique history. We also need to be aware of the implicit pressure on behalf of on providers because of systems. For example, providers need to understand why there could be a push for LARC methods from an economic standpoint. You know, overall, what are the metrics of success that a hospital might be using or a system might be using that result in sort of this bias in counseling. And ultimately ensuring all access is equitable, including LARC removal when desired. Next slide. How do we take these principles and this knowledge that we have about the patient experience, about where we should shift our emphasis in counseling and provision into research? So really, I think we start with acknowledging our dark history in reproductive health care. Really, patience and understanding is essential when working with certain communities, specifically Black and Indigenous people of color communities, and their potential fear and hesitation of research and experimentation. We need to think about recruitment of racially diverse populations into our research and why it's important. Diversifying research teams will also really help in increasing representation and increasing representation of our specific populations in the research itself, as well as in uptake of the research. We should work with community leaders within trusted spaces as we think about research and dissemination, and ultimately use culturally competent and appropriate research frameworks when designing our work, and prioritize subjects and patients' needs and comfort first. Next slide. Next slide. So I'll just finish with our, this is obviously very shifted gears from the previous talks, and I think it's just important to keep this centered as we move forward with our work. So our desire is really to create and optimize IUDs and increase their acceptability and access. And in doing so, we need to keep in mind the individual and community experience of our patients and what we have learned from this research thus far in order to balance equity with access. And I'll thank you for the opportunity. I'll turn it over to Dr. Curtis now.
Thanks, Sadia. That was really a wonderful presentation. And thank you, Allison, and the organizers for the invitation to join this final session of the IUD Symposium today. Hi, everybody. I'm really delighted to talk with you today about updating CDC's contraception guidelines. We've got lots of questions recently about whether and when CDC will be updating these guidelines. And so I'm excited to talk with you today about our process and our planning activities. Next slide, please. So CDC's Division of Reproductive Health produces evidence-based contraception guidelines for healthcare providers that are adapted from global guidelines from the World Health Organization. And these guidelines are intended to assist healthcare providers when they counsel people about contraceptive method choice and use. The U.S. Medical Eligibility Criteria for Contraceptive Use, or USMEC, provides recommendations for the safe use of contraceptive methods by people with various characteristics and medical conditions, such as hypertension, diabetes, or being postpartum. The USMEC currently includes over 1,800 individual recommendations for more than 120 conditions and characteristics. The companion US Selected Practice Recommendations for Contraceptive Use, or USSPR, provides recommendations on common contraceptive management questions, such as how to start a contraceptive method, what exams and tests are needed before starting, and how to manage side effects or other management questions. Next slide, please. Before I talk about our process, I want to start with a broad overview of evidence-based guidelines activities. So the center circle in this diagram represents the activities that go into developing and updating evidence-based recommendations, which is what I'm going to focus on today. But this is just one part of the guidelines process that's centered in the midst of these other critical activities that you see on the left and the right of the diagram. On the left side, you can see the research loop. So research and evidence, of course, are the backbone of the guidelines process. But equally important are the gaps that are identified in the guidelines process. And then as we identify those gaps, prioritized research can be conducted and fed back into the guidelines process to strengthen those recommendations. On the right side, you'll see dissemination, implementation, and evaluation activities. And as we all know, there are many steps and sometimes large and complex barriers to go from making a recommendation to effectively implementing that recommendation. We also need to evaluate how the recommendation works in real life, and that evaluation information can be fed back into the guidelines for improvement. Next slide, please. So CDC has worked with WHO's Department of Sexual and Reproductive Health and Research since the 1990s on developing and updating global recommendations for contraceptive use. WHO determines these recommendations based on the evidence and within the framework of reproductive and sexual health care as a human right. These global recommendations are meant to be used as a reference that countries or institutions can adapt to meet the needs of specific healthcare and service delivery environments. So in 2010, CDC first adapted WHO's MEC for use by healthcare providers in the United States and followed with the SPR in 2013, with the last comprehensive US update in 2016. We generally follow WHO guidance updates, and we consider whether to adopt the WHO recommendation for the US or whether there's something specific about the US context that would lead us to adapt the recommendation for best implementation by US healthcare providers. CDC works together with WHO to keep guidance up to date based on the best available evidence. And we use a system called the Continuous Identification of Research Evidence, or CIR system, to review newly published evidence on a weekly basis. We use that new evidence to update systematic reviews, and then we use those systematic reviews to consider when a recommendation might not be reflecting the evidence anymore, and we should consider an update. Complete reviews are usually done about every 5 years, but interim updates may also be developed when new evidence warrants or based on new WHO guidelines. And you can see at the bottom here an example of our recently released recommendation on self-administration of subcutaneous DMPA. Finally, an essential component of developing guidelines is participation by a wide range of partners to ensure that the recommendations are feasible and that they really meet the needs of providers and their patients. Next slide, please. So now I want to turn to CDC's established process for updating the USMEC and the USSPR, which includes 4 steps that you can see here, and I'll go through each one of those. So next slide, please. So the first step focuses on determining the scope of the update. So it's not possible or even necessary to review every individual MEC and SPR recommendation every time we do an update. And we want to prioritize potential changes or additions that will have the biggest impact. For existing recommendations, we look at new evidence that might have changed the overall body of evidence for a recommendation or might provide new information on a specific aspect of a recommendation. Since our recommendations are adapted from WHO, we also look at recent WHO updates and we review several sources of external feedback. We also consider new recommendations, including recommendations for new contraceptive methods, additional MEC conditions, and new practice recommendations to be added to the SPR. For new recommendations, we consider whether there's a body of evidence on which to base the recommendation, the impact of potential recommendations, external feedback, and whether there is a new WHO recommendation on the topic. We also are really focused in thinking a lot about how to take patient perspective into the guidelines process and really starting at this very beginning stage of the scoping. While the guidelines themselves are for healthcare providers, are there ways that we can best frame, determine, and present the recommendations so that they can assist providers in providing patient-centered and equitable family planning and reproductive health care? Next slide, please. Once we've determined the scope of the update, the next step is to update our systematic reviews for existing recommendations that we are considering revising, and to conduct new systematic reviews or find published systematic reviews that will support any potential new recommendations. We follow current standards for systematic reviews and meta-analyses. We consult subject matter experts as needed, particularly for new medical conditions. We send out our systematic reviews for peer review before we present them to a recommendation meeting. And then we publish those systematic reviews so that they are available. Next slide, please. So the third step is to convene a meeting of external participants who present a wide range or represent a wide range of expertise and experience. During the meeting, we present the evidence in the systematic reviews, and the participants provide their individual perspectives on potential recommendations. So during that meeting, we of course consider the evidence from the systematic review, but we also consider the benefits and the harms of a potential recommendation, as well as the values and preferences of providers and patients. And we think about those values and preferences both around general family planning issues, as well as around recommendations for the specific medical conditions and characteristics or for specific practices. And considering values and preferences can be especially important for recommendations where there's a lack of sufficient evidence and for which patients might make different decisions. And as I said before, for the WHO recommendations that we're considering, the key question for the US is whether we want to adapt or adopt the WHO recommendation. Next slide, please. Finally, CDC considers all of this information and determines the final recommendations. We publish updated versions of the guidance in CDC's MMWR publication and update provider tools and job aids. And then we begin the process of dissemination, implementation, and evaluation, and identify and publish the research gaps that can feed into the next recommendation update. So CDC intends to start the update process later this year, and hopefully that will lead us to release updated guidance in 2023, which is not as far away as it sounds. Next slide, please. So thank you very much. I look forward to your questions and discussion at the end of the session. And we really are looking for your feedback as we head into this planning session for the next update. Thanks very much. And I will turn it over to Lisa.
Thanks, Kate. And thank you to the organizers for the honor of presenting the symposium finale. Over the past year, we've heard from many of the giants in domestic and international contraceptive research, highlighting where we are. Now we all need to work together to develop a future research agenda. My biggest disclosure is that I'm an idealist and truly believe that research can help technology to reach its full potential and positively impact lives of women globally and individuals globally. Next slide, please. So I've tried to summarize my thoughts around 3 central issues to capture many of the topics explored during the symposium: IUD access, optimizing client-centered care, and new technologies. First, let's recognize how far we've come. IUD use is expanding, with increasing use globally and in the US, despite a challenging history. This is encouraging, but let's not rest here. This word cloud summarizes multifactorial themes raised that influence IUD access. In earlier sessions, including those with Jennifer Amico, Diane Green-Foster, and Gillian Dean, many barriers to IUD use and access were identified, such as costs, delays in getting appointments, and providers' unwillingness to remove IUDs if they didn't place them. Key questions we need to address through research include, can we make placement and removal easier and reduce costs? There's an interest in self-removal, with some individuals more likely to uptake an IUD if self-removal was an option. Yet self-removal is often not provider supported or encouraged, with Google and YouTube often the source for patient information. How can we better support self-removal and fill gaps in our understanding on self-removal counseling in ways to enhance access and success? Can telehealth expand access and remove barriers to care? Offering remote counseling or side effect management via telehealth may reduce barriers to care and enhance capacity for challenged healthcare systems. COVID has accelerated the push towards self-care and alternative methods for healthcare delivery. Hybrid models with telehealth have been particularly useful during the pandemic. However, moving forward, can we leverage these methods to expand services and overcome access challenges? And can implementation strategies ensure equity in a rollout of a telehealth system? Moving to training. Training providers for insertion of IUDs in some settings still remain a challenge, especially in regions with limited access and overburdened healthcare systems. It's not uncommon that after IUD trainings, providers lack ongoing support to continue with confidence offering these services and encounter great difficulties in implementation of the service of IUD placements. Without local champions, the efforts to increase access can quickly revert. Testing and improving on alternative training platforms and advancing technologies to simplify insertions can reduce the pressure by similarly, the demand side must be built in parallel to support the growth and maintaining of access. Can we expand and sustain distribution channels to increase access in new settings? Further, affordability of IUDs is central to improving access. This was a great achievement with the first copper T, with costs of only $1 for over 10 years of coverage. Although more recent leave-in adjuster IUDs have sought to reduce the cost of goods, the cost of the copper IUD is unlikely to be matched. That said, the cost of the device is only a small aspect of the overall costs associated with care. In many healthcare systems, setting placements and removals can cost a high amount and often be unattainable despite insurance coverage, and often translate to extensive costs that can be inhibiting patients from utilizing an IUD. In this area where advocacy is imperative, research can demonstrate a cost benefit and a language often most clearly heard by policymakers and insurers, and can continue to provide essential evidence to support the service and provision in diverse settings. We have heard the importance of advocacy for driving change and the value of research in supporting effective policy change. This was highlighted during the session last month with Alina Selganikoff, Christina Gemsel-Danielson, and Michelle Moniz. Future research must be informed by and inform advocacy to effectively enhance policies to reduce barriers and ensure we are able to maximize access for all eligible populations. A central question is, how can we maintain equity and justice as central tenets to the efforts to increase access? Next slide, please. The learning agenda must enhance client-centered care to better meet the needs of individuals. Counseling, educating, and recognizing the unique needs of different populations, including adolescents and transgendered individuals, need to be addressed. The research agenda must address questions such as, how do we keep the client in the center? How do we reduce provider stigma and perceived coercion? What tools are needed to improve care? How do we redesign care to support clients better? Can counseling tools better balance the discussion of effectiveness with other key features that are sometimes even more critical to an individual's choice and method? Here's an opportunity to highlight secondary benefits that can be a driver for use. How can we measure acceptability, success, and improve on it? Can we design better tools to identify, support, and manage side effects? This is an area where self-care can be strengthened, tools enhanced, and supported individuals who may desire to receive care differently or have different motivations to use besides contraception. History has stained the landscape. We must integrate our understanding of these past experiences and perceptions to address potential and future user needs. Unfortunately, stigma and coercion is still perceived, as many of the prior presenters in Sadia have highlighted, with patients feeling resistance to IUD removals, implicit pressure, racially based discrimination, and disregard of patient voices. These perceptions can lead to long-term consequences on health care mistrust and future contraception non-use. How can we, as providers and researchers, with self-reflection, adapt our approaches to reduce stigma and coercion? Next slide. Lastly, the area I'm particularly excited about are new technologies. We've heard earlier today about new devices, including flexible shape memory Nitinol frames, smaller profiles, and lower copper loading IUDs. There are also different shapes, such as the intrauterine ball and new inserters that can reduce the training requirements for safe use and expand success in settings such as postpartum insertion. Many critical questions need to be addressed with emerging technologies, including, will these new technologies enhance the user experience and reduce side effects? Can we improve on the safety and reduce costs? Can placements be made easier and safer? We have heard encouraging data for expanding the indications for current IUDs, such as the leave-in adjustable IUD as emergency contraception. Also, future multi-purpose prevention technologies that may offer integration of HIV prevention services or other APIs directed toward enhancing bleeding control without androgenic side effects. Treatment for endometriosis or fibroids. Will regulatory burdens associated with multiple indications for MPT IUDs slow progression of these IUDs to the market? Are there other indications to explore for MPT IUDs, such as STI prevention and vaginal health? And let's not forget about the dozens of IUDs available globally that will never be able to enter the US market, challenged by the costs of clinical trials in the US required by the FDA and no EMA reciprocity permitting approval. Many IUDs have been on the market for decades with exceptional safety data. How can we be confident that newer technologies do not lose momentum short of bringing their technologies to all global markets, including the US? As the IUDs currently are incredibly safe with high tolerability and continuation, will new methods be able to go beyond non-inferiority to demonstrate superiority? Can the market support IUD expansion and new methods? Will policies provide expanded coverage and support access for new methods that may have slight incremental improvements on prior, more cost-effective options? Will insurance coverage limit access to non-generics? This is an issue of advocacy that we need to think about as we push the future research agenda. And critical to this point, will there be sufficient funding to support these developing technologies? And lastly, with advancing technology, we may also soon be able to have tools to improve our counseling and personalize our care. We know that different individuals have different experiences with use of contraceptive methods. Our counseling is limited based on our inability to predict user experiences with prior studies, often relying on insensitive tools for analyses. Some bright hope may be found in pharmacogenomic analyses to aim in our ability to identify individual genomic variants that may be associated with different side effect profiles, and metabolomics and transcriptomics to help us understand the different responses that may underlie the effects and help us target treatments. With advancing technologies, we may soon be able to personalize medicine. Next slide. To conclude, I am hopeful. Let's get back to the 1970s and early 80s, where in the US, many had written off the use of the IUD, yet some persisted and believed IUD still had an important place in women's health. As we define the future research agenda, we must continue to hold onto our idealism. Seek what we want, ensure equity and justice are central drivers. Focus our efforts on individuals and search for tools to improve and broaden the care for those we provide services to. There are many challenges from the past that we need to overcome in gaining trust and expanding the impact of IUD technology across borders and into all populations. I'm encouraged by the work that is being done, yet believe there is so much more to do. My hope is that for many of you, we can help shape this agenda moving forward, work for it, and fight for it. Thank you.
Oh, thank you, everybody who spoke today, and thank you, Dr. Haddad, Dr. Curtis, and Dr. Haider for ending the panel with such lovely presentations and kind of regrouping about what has been learned during the year. We do have a couple minutes for questions. I'm going to stop sharing my screen, and if you have questions that you can put in the chat for either the speakers before or the speakers at the end. I think, because we didn't have enough time at the beginning. And I encourage the speakers, if you're still present, to turn on your cameras if you can. While we're waiting to see if any questions come up, I know that we had a question in the chat from Dr. Hubacher, which I think is for Dr. Haddad, about just self-removal. And should we seek to improve efficacy before we advocate for this removal option? I don't know if you have any thoughts about that.
So there were earlier speakers last year that addressed many of the challenges with self-removal, and there were, the data seemed to suggest that only about 20% of individuals in those studies were successful with self-removal. So I do think we need to study further how we can improve on self-removal before we broadly advocate for it. But we know that individuals are going online and seeking alternatives to figure out how to remove the IUDs themselves. So being prepared and developing the appropriate tools in order to really support self-removal is going to be imperative.
Great, thank you. Kate, I had a question for you about how are we doing better about improving that patient perspective in the guidance that we bring forth, given all of the difficulties that we've had with hearing those voices and how important they are for clinical guidance.
Yeah, thanks, Allison. That's a great question. And it's one that we've really been thinking a lot as we're starting to plan for our next update. As I talked a little bit, I think it's really important to try and figure that out at the beginning as we're even scoping our update. Are we even asking the right questions? Are we talking to the right people? How can we help, even though that our target audience is healthcare providers? What can we do with the recommendations that can best help providers to take that patient perspective into account, to provide patient-centered, equitable family planning care? Part of that we can do through values and preferences. I think the values and preferences concept is a good one for this. It's difficult to get those values and preferences. Ideally, we would have lots of qualitative studies from all kinds of populations that would ask people about their values and preferences about family planning in general and about specific issues. There's getting to be some more of that, and I would direct people to, there's a series of literature reviews on values and preferences around family planning that's being published in contraception, but WHO and Johns Hopkins are the lead authors for that. So that's one thing we can do. We also can use through the GRADE process, which is an approach for creating evidence-based guidelines. There's something called an evidence to decision-making framework. And I think using that framework can explicitly take into account not just the evidence, but things like values and preferences, benefits and harms, equity and human rights, feasibility and costs. So that's a way to make sure that we're really walking through all of those issues and taking that into account. And then one other thing that we're considering doing specifically for this update that we haven't done before is putting a notice in the Federal Register, which sounds very governmenty, and it is, but it's the government's way of getting information out. So we will probably solicit for public comment for our update, which is not going to it's going to get us a lot of great stuff. It's probably not going to reach very far down into patient perspective. So we do need to think more about that. And I would love to hear other people's perspectives or experiences about that as well. But we're certainly thinking about that.
Great, thanks so much.
Thanks.
I am recognizing that we are at the hour, and one of the tenets of virtual meetings is to make sure to end on time. I see a great question by Dr. Westhoff, who also is part of the organizing and lead for the committee of the symposium. And I think, some of the chats have been published afterwards. And so I'll encourage the speakers to actually write what is your personal biggest priority for research and implementation, and then hopefully we can publish that as part of the the webinar today. But I do want to thank everybody so much for attending today, and for those of you in the future who watch this, if I actually recorded this appropriately, since I was put in charge all of a sudden of the recording and managing the chat. So I apologize if this actually didn't get recorded. And just thank you again for attending, and for all the speakers in the coordinating committee. It's been quite a year, but we're at the end, and we really appreciate you all being here, and hope you're all doing well and your families are well.
Thank you, Allison, for stepping into the breach and hosting the meeting. And thank you again to Regine for stepping into filling in Vivian's shoes on very short notice. We really appreciate everyone's efforts. This has been a great series, and the organizing committee thanks you all for attending, because it was quite a challenge to adapt from what we'd hoped to be a wonderful in-person meeting to these rather unusual series. But thank you for attending, and we look forward to seeing you all in person at some point in the future.